Okay, hello everyone. Let us get started for today. I hope my screen is visible to everyone. Perfect. Hmm. Okay, welcome everyone to week 6 PMRF-NPTEL series on molecular biology. As you would know that these are problem solving sessions that are held by a PMRF fellow such as myself. And in this particular series, we go through the previous year's quizzes of the same course so that we have a better understanding on how to tackle the course, get better understanding of the concepts behind them, and also how to solve the quizzes in the exams as well. So, brief introduction for myself. My name is Anuvab Chatterjee. I'm a PMRF fellow at IIT Kanpur, and I am taking this course. And this week, we are going to deal majorly with transcription in both the prokaryotic and eukaryotic system, as well as look at how transcription is regulated. So, does anyone have any questions regarding this week's videos itself? Then we can tackle that first. Otherwise, we will go right into the questions. Okay. If there are no outstanding queries, so we'll go into the question. So how we do this is that I will show you a question from the previous year's run of the course and ask you to try and solve it. And then we will discuss whatever the concept behind the question is and then answer them together. So the first question for today is, during transcription, only one strand is transcribed, so that the transcribed sequence is identical to one strand of the DNA, which is known as the template strand, the antisense strand, the coding or sense strand, or the non-coding strand. You can put whatever you think is the right option in the chat as A, B, C, and D. Okay, Rupsa says it is A, the template strand. Jagannath says it is C, the coding strand. Anyone else? Chanda says it is A, the template strand. Anyone else wants to try and answer this? Okay, so let us look at it. So I will use this question to give you a brief crash course on, or a brief introduction to transcription, so we have a good understanding going forward as well. So transcription we have seen is the process of making a RNA from a known DNA stretch. So this is known as transcription, DNA to RNA. This is all part of the central dogma of life. We know that there are three different processes that occur. DNA forms DNA, which is known as replication. DNA forms RNA, which is known as transcription. And RNA is used to make protein, which is known as translation. Three different processes, and obviously the entire idea is the inflow of information is unidirectional from DNA all the way to the protein. DNA essentially acts as the blueprint, and protein actually act as the actual biomolecules that do various processes. So making of membrane, metabolism, division, cell division, everything is done by proteins, but the blueprint for the proteins is stored in the DNA. But this information stored in the DNA is first transcribed to an intermediary molecule known as the RNA, and then that information is transferred to the protein. So we are going to deal with transcription, which is DNA, the flow of information from the DNA molecule to the RNA molecule, which is done by a enzyme known as DNA-dependent RNA polymerase. So there are one or two things I would like you to know. The first being that whatever stretch of DNA codes for a RNA is known as a gene. So you would have heard what a gene. The formal definition for gene is any stretch of DNA that can code for a functional biomolecule. It could be that it is coding for a RNA or it is coding for a protein. So RNA to protein. So there are different types of RNAs present. The one that gets translated to the protein is called the mRNA or the messenger RNA. There are other RNAs as well, such as tRNA or rRNA or microRNAs or snRNAs. All of these RNAs are not converted to the protein but have functions of their own in their RNA form itself. So these are non-coding RNAs. So they do not code for a protein, but they function as RNA. But all of them come from a gene present on the DNA, which transcribes this functional biomolecule, which could either be a RNA or protein. But today we are going to look just at genes in the DNA that code for mRNAs, which then are translated to proteins. So this is our topic for today. So what exactly is transcription? So transcription is the process of DNA getting converted to either rRNA, tRNA, mRNA, or microRNA or snRNA. So any kind of RNA being formed from DNA is known as transcription. So this you can remember it as something like this, that since we are going from nucleic acid, which is deoxynucleic acid DNA, to normal RNA. So this is called transcription, as if you are looking at a language and you are taking rough notes as DNA and you're transcribing into your fair notes as RNA. So it is known as transcription. The next step, which is taking the information from the RNA to make a protein, is called translation, because you are going from the language of the nucleic acids to that of the amino acids or the proteins. So DNA to RNA is called transcription. And the ones that we are interested in are mRNAs, which are the messenger RNAs that are used to carry information to the proteins. So synthesis of RNA from a DNA template with the help of a DNA-dependent RNA polymerase is known as transcription. It always occurs unidirectionally from the 5' to the 3' direction. So RNA polymerization occurs from the 5' to 3' end, and the segment of the transcribed DNA is known as the transcription unit. So this here is an example of a transcription unit. Now, very simply, it contains the promoter, the RNA coding sequence, and the terminator. We will see what each of these does in further slides. But one important fact that you should know just now is that in eukaryotes, the transcription unit is monocystonic, which means that one transcription unit will code for one protein, or one transcription unit will just code for one RNA. Whereas in prokaryotes, one transcription unit can code for various different RNAs. So there could be multiple RNA. So gene 1, there could be a gene 2, gene 3 inside one promoter and terminator stretch. And they can in turn give rise to different proteins, protein 1, protein 2, and protein 3. But in eukaryotes, that does not happen. In eukaryotes, you only have gene 1 in between a promoter and terminator, and that only makes one protein, protein 1. So that is why eukaryotes are known as monocystonic, and for prokaryotic transcription unit is polycystonic. Some details about the transcription unit itself. So you have the promoter. Promoter is a stretch of DNA on which our RNA polymerase would bind. So it is the stretch of DNA on which actual the RNA polymerase enzyme that will do make the mRNA would bind. And then you have the first DNA uh, base pair or DNA nucleotide that actually the RNA polymerase reads to make the first RNA base. So that is called the start site. The first transcribed DNA nucleotide is known as the start site or the +1 site. So it is the first base from where transcription starts and is called the start site. And the polymer starts moving from this point along with the template, so it will move in this direction and synthesize RNA up to the upstream terminator sequence. So looking from the start site, you either have sequences which are upstream, so towards the left, where towards the promoter, and even more further upstream towards other DNA sequences, and downstream, which would be toward downstream would be towards the template. So in this direction, towards the right and towards the left. So it upstream is the non-template nucleotide where the 5' end or minus direction sequence before the start point. So any sequence that is before the start point is upstream, and any sequence which is after the start point towards the 3' direction is called the downstream. So DNA is a double-stranded structure, and during transcription only one strand is transcribed. So during transcription, the DNA opens up as a bubble, which is known as the transcription bubble, and only one of the strands is actually synthesized. So since RNA, as we have said, can only be polymerized from the 5' to the 3' direction, the synthesizing the RNA polymerase actually reads the non-coding strand, the strand that is running from 3' to the 5' direction. It reads this strand. And depending on the complementarity between DNA and RNA bases, so A binds to T, U binds to A, G binds to C, C binds to G. It keeps on adding new nucleotides. And this occurs in the 5' to 3' direction. Thus, you have two different strands here. One, which is known as the non-coding strand, on which the RNA polymerase actually sits and reads to make the mRNA. And this strand is also known as the template strand or the antisense strand. The other strand, the strand which the polymerase actually never touches, is known as the coding strand. So once you make the mRNA, so whatever the sequence of the mRNA is going to be, it is going to be identical to the coding strand because it is the mRNA is complementary to the non-coding strand. So for example, so if our coding strand was ATG and something else, so our non-coding strand will be TAC. TAC. So our non-coding strand will be TAC. When the mRNA gets synthesized, it is complementary to the non-coding strand. So the bases are A, T, or A, U, G, and C. But this sequence of the mRNA is completely identical to the coding strand because obviously there's only going to be one complementary base corresponding to the non-coding strand. So thus the strand above, which the mRNA polymerase never touches, is known as the coding strand, and the sequence is identical to the RNA being synthesized. So the lower strand on which the polymerase sits is known as the non-coding strand or the template strand or the antisense strand, and the upper strand is known as the coding strand or the sense strand, which has identical sequence to the RNA being synthesized. Now a bit more information about how transcription occurs. So this is a diagram showing how transcription is occurring. So we have seen there is a promoter on which our RNA pol first binds. Then we have a stretch of DNA which the RNA pol looks at, reads, and makes the mRNA or the RNA. And then there is a terminator sequence at which the RNA synthesis will stop. So there are four different steps. Template binding. So RNA pol goes and sits on the DNA. Chain initiation, where the first RNA uh, base is added. Chain elongation, where the polymerization of a longer RNA occurs, and termination, in which the polymerase stops doing its job and the RNA is released as a newly synthesized RNA strand. So, binding of RNA polymerase to template DNA and chain initiation. DNA duplex can be opened so that the RNA pol can approach to a single-stranded DNA template. So, as we have said, that RNA polymerase sits on the single strand. It does not do transcription on both strands. Efficiency is inversely proportional to the Tm. Tm, you will remember, is the temperature at which the strands melt. So obviously, a AT-rich region where there are only two hydrogen bonds between A and T is easier to break than a GC-rich region where there are three bonds. So the breaking of or the opening up of the DNA occurs at the AT-rich region. So the AT-rich region is good for melting of the duplex, easy to create an open promoter complex. And what exactly so this is what the promoter is. The promoter is the region where the two strands starts to separate from each other, and this particular region is detected by the sigma factor of the polymerase, and then other subunits of the polymerase, the alpha and the beta, sit on the sigma factor to form the hollow enzyme which actually will do the transcription. So this is known as the so this is how the RNA pol actually sits on the DNA. Next, it will start doing transcription, which is done by the uh, open complex. So the closed complex is converted to the open complex by melting DNA at the -10 region. This -10 region is important. So all genes have a Tata box sequence, which is TAT. So essentially written down as Tata. And this is, as we have said, AT-rich region, which is easy to open. So RNA pol sits here and unwinds the DNA from all the way to -55 to +1. +1 is the side where the first base will be added, and -55 is all the way upstream. So entire thing opens up, and then new nucleotides are added in the RNA chain. So this is known as the open complex. Then chain elongation takes place. Essentially, RNA polymerase moves along this non-coding strand or the template strand in the 5' to 3' direction, and it looks for base paired RNA molecule bases and keeps on adding new and new RNA bases to the elongating RNA chain. So RNA synthesis is carried out by the transcription bubble due to transient separation of the double-stranded DNA into single-stranded DNA. Transcription takes place at the template DNA strand. And finally, we have the RNA chain synthesis occurs from 5' to 3' end, and always around 12 to 14 nucleotide stretch is opened up by RNA polymerase as it moves along the template strand. And nucleotides are obviously added in the 5' to 3' direction from the 5' phosphate to a 3' OH, and this keeps on going till we reach the terminator sequence. So during transcription, only one strand is transcribed so that the transcribed sequence is identical to the other strand, which is known as the coding or the sense strand. So the one strand that is transcribed is the template or the antisense or the non-coding strand, and the sequence that is transcribed as RNA is identical to the coding or the sense strand. Any questions? Think for a bit. Let me know if you have any questions. Okay, it seems there are no questions, so we'll move forward. The Rho protein is a hexamer ATP dependent helicase which is involved in Rho dependent blank. The options are replication, transcription elongation, translation, or termination of transcription in prokaryotes. You can put in whatever you think is the correct option using ABCD. Jagannath says it is D. Anyone else? Bhavna also says it is D. Termination of transcription in prokaryotes. Anyone else? What is the Rho protein involved in? Okay, so let us look at it. So we have talked a bit about how initiation takes place, elongation takes place, and the final thing is termination. So how long will the polymerase sit on the DNA and keep on adding uh, more and more RNA bases? So how long will the mRNA stretch? So that is determined by the termination sequence. So this is the point at which the RNA polymerase stops adding nucleotides to the RNA chain and then moves off the DNA segment. Once it moves off, the RNA is dislodged and this goes away to do translation. So this is known as the termination or the chain termination. So when the polymerase stops adding nucleotides to the RNA chain, it releases the completed product and the RNA chain is free from the terminator sequence. During termination, all the hydrogen bonds break between the RNA DNA hybrid, which was being formed to, so there was RNA DNA hybrid hydrogen bonding being formed as the RNA polymerase kept on adding new nucleotides. But once chain termination is site is reached, the polymerase falls off, and this entire hybrid moves out from the duplex, and the DNA can bind together again. And when the RNA chain is separated from the DNA chain, which again forms the duplex. The site at which the enzyme stops adding the nucleotides is known as the chain termination site. And there are two types of termination mechanisms. One is the intrinsic termination, and the other is the road de facto dependent termination. So let us look at it. The first is intrinsic intrinsic transcription termination. So intrinsic, we know it means that it occurs internally. You do not need any external factors for it. So how does that work? So the transcription stop site is a stretch of specific nucleotides. So, so it is a GC rich region. So the GC rich region here we can see, and it is made in the way that normally it forms a duplex to the other strand. But once it forms a single strand, which occurs during transcription, it can actually form a hairpin loop. So the chain just opens up and binds to other sites in between and then closes. So this happens with, so you can see TTA, TCC, and CCT, these are complementary to each other. So essentially, if you ever open it up, these two now have the opportunity to base pair, and this is extremely common in RNA and also DNA, and these are known as secondary structures of DNA. So the same strand can just bend, loop, and then form hydrogen bonds with themselves, forming this hairpin loop structure. So here, these parts are all hydrogen bound to each other, and essentially this forms a obstruction for the RNA pol, which is going trying to move in this direction because there is a huge obstacle in the road that it was following. So it is a GC-rich hairpin followed by 7 U residues. So there are A residues on the coding strand or the template strand, which means here U's should be incorporated in the RNA. So RNA DNA hybrid requires forces for holding elongation complex together, and thus when the hybrid gets detached, it collapses and the elongation complex, which causes the termination. So essentially, when they reach the termination loop, followed by a lot of use being required, this causes too much strain on the RNA polymerase elongation complex and it just falls off. In this type of termination, dissociation of polymerase occurs by destabilizing the attachment of growing chain to the template. During this process, the hairpin structure is formed by the transcript via complementary base pairing. It includes palindromic sequences. This stem structure includes GC followed by a U-rich area, which we see right here. GCs, a lot of Gs and Cs, followed by a U-rich area in which the RNA needs to be added. So once the polymerase reaches this site, it completely slows down and is not stabilized enough and it falls off, and that causes transcription to stop. The other kind of transcription regulation is known as row-dependent transcription regulation. So in this, I will quickly summarize is, so this is our RNA pol, which is moving along the DNA single strand. And here is our terminator sequence. Now, the row protein is a hexamer ATP-dependent helicase, which can bind to a upstream sequence known as the RU3 or RUT sequence. So, row protein will come and bind somewhere upstream to the already transcribed region that the RNA pol has transcribed. So it binds right here. And now essentially RNA pol and Rho chase each other. So RNA pol is running towards the DNA strands and Rho is following along just behind the RNA polymerase. The moment the RNA polymerase reaches the termination sequence, which we have already seen, is made out of a hairpin loop, which causes the RNA polymerase to stall. So it stops here, and Rho essentially reaches up and pushes it off the DNA segment. So essentially, Rho crashes into RNA polymerase, destabilizing it and pushing it off the DNA strand, and thus, which completely prevents any more polymerization to occur. So in this case, RNA poly is actively pushed off the DNA segment by the Rho protein, which is following up. Essentially, you can imagine two trains traveling, one from behind, one is going. You see obstacle, the first train stops and the second hits it from behind, and the first just falls off. And this essentially causes transcription to completely terminate. So, yeah, this is the same thing. And then after RNA polymerase is pushed off, the entire RNA strand is released, and thus you have made a new RNA strand, and transcription is complete. So Rho protein is a hexamer ATP-dependent helicase, which is involved in Rho-dependent termination of transcription in prokaryotes. Any questions? Okay. The next question is, during post-transcriptional modification, splicing mechanism of both group 1 and group 2 involves similar steps of two blank reactions. So this is transesterification, hydrolysis, methylation, or hydroxylation. You can put in what you think is the correct option in the chat. Okay. Jagannath says it is C-methylation. Anyone else wants to try? Bhavna says it is A, transesterification. Let us look at it. So now we are going to talk about post-transcriptional modification. So you have already transcribed your gene and it has made a mRNA, and now you're going to make changes to the mRNA molecule that just came out. So these kind of post-transcriptional modification that we are going to discuss all happen in eukaryotes. It does not happen in prokaryotes. The major reason being that in prokaryotes, the genomic material is present directly in the cytoplasm. So the moment a mRNA strand is synthesized, the ribosome sits on the mRNA strand, and essentially transcription and translation occur on the same strand. As it is being made by the RNA polymerase, the ribosome keeps on making the protein by sitting behind on the newly synthesized RNA stretch. So, in that case, there is no question of post-transcriptional modification. But in case of eukaryotes, the genomic material, the DNA, is in the nucleus. Once the RNA is made in the nucleus, while it is being transported from the nucleus to the cytoplasm, where the ribosomes are present and they will start doing translation, they are actually subjected to multiple modifications. Majorly, they help in longevity of the mRNA so that it can be used again and again for making proteins, and we will see how that occurs. So the first post-transcriptional modification is the addition of the 5' end cap. So RNA by itself, you would know that the reason RNA is not the chosen genomic material is because RNA is very unstable. And further, on top of being unstable, there are multiple RNases, which are enzymes which chop up RNA present in the cell, which does not want any rogue RNA to go around and start translating proteins for itself, because that is how viruses act. Many viruses are made out of RNA, or they make new RNA that are not the cell's own RNA, and the viruses take over the cell's own machinery to make proteins for its own benefit. So the cell always monitors the situation by producing RNases, which will go up and chop any rogue RNA going around. There are two kind of RNases. One is called endonuclease and exonuclease. Are ones that start acting from ends of the DNA, either from 5' end or the 3' end. Endonucleases attack the middle of the RNA. So essentially, the two major modifications that we'll discuss are made in order so that the RNA is not affected by the cell's own exonucleases, so that the enzymes which start chopping off RNA from both ends actually cannot function very well, which essentially increases the lifetime of the mRNA. The first thing that occurs is called 5' capping. You want to prevent enzymes such as the 5' RNase from recognizing this as a 5' like this as an open 5' end. So how does that occur? So in eukaryotic cells, mRNA is inherently unstable at the end, so they need to be modified. The first kind is known as 5' capping. So what does 5' cap does? It what it does is that at the end of the 5' capping process, a slightly modified guanine residue, so a slightly modified GTP GTP is added, and this modification is at the 7' end, the seventh position in the guanine nitrogenous base, where a methyl group is added. So this is looks very similar to a normal GTP, but on the seventh position of the nitrogenous base guanine, there is a methyl group added. And this is attached backwards in a 5' to 5' linkage. So any kind of polymerization we have seen so far, always the polymerization has occurred from the 5' to the 3' end. But here, in this case, the attachment is from the 5 prime to the 5 prime end. So the attachment is going to be from the 5 prime to the 5 prime end. So the enzymes cannot recognize this kind of linkage, and thus 5 prime exonucleases do not work on mRNA. So essentially, you are stabilizing the mRNA. So it is attached backwards by a 5 prime to 5 prime linkage to the triphosphates of the first transcribed base. The capping reaction includes condensation of the GTP with triphosphate at the 5 prime end, followed by methylation of guanine at the N7th position. Further methylation occurs at the 2 primes with second and third nucleotides adjacent to the cap to make this even more secure from 5 prime exonucleases. So this is called 5 prime capping, and this is very important for translation as well. Next, so you have protected the 5 prime end. Next, you want to protect the 3 prime. The 3 prime end is protected by something known as a poly-A tail. So there is an enzyme called poly-A polymerase. So PAP recognizes a specific site on the mRNA's end to the 3 prime end, which is known as the poly-A site. PAP sits on it and keeps on adding as many A's as it could possibly find. So it is a template independent RNA polymerase. It does not require a template. Its only job is to add A's, as many as it can. And by the end PAP is done, you have 80 to 250 nucleotide stretch of A's being added to the end of the mRNA. So again, this is done for protection against 3 prime exonucleases, which will start chopping up the mRNA from the 3 prime ends. Because there is this huge of a poly A tail, by the time the nucleases actually reach important information, the mRNA has already done its job. So this is known as poly A tailing of the mRNA. And this is a very important and what can I say? Unique characteristic of eukaryotic mRNAs. Eukaryotic mRNAs always have a poly A tail, and this fact is used for various molecular biology approaches. And finally, there is something known as splicing. So, they on the DNA. So we have seen that there was a promoter region, there is a RNA coding sequence, and there is a termination region. But this, if you just look at the DNA, all parts of the DNA are not converted into a mature mRNA. There are sequences in the DNA which are removed, which are not present in the final mature mRNA, which are called introns. Whatever is present on the DNA that remains on the mature mRNA is called an exon. So essentially, introns are these sequences which are non-coding and they are removed when the mRNA matures. So introns are non-coding nucleotide sequences within a gene that don't code for protein and don't appear in the final mRNA molecule and are removed by a process known as splicing. Protein coding sequences of the gene known as exons are interrupted by introns in eukaryotes. So there are exons followed by introns, followed by exons, followed by introns in the alternate fashion in the DNA. DNA gets transcribed to the mRNA. mRNA is then spliced so that the introns are removed, and finally, mature mRNA that is translated to a protein only has exons. So introns come in 4 different varieties, group 1 and 2, in which there occurs something known as a self-splicing intron. So these, once the mRNA is formed, these introns just say goodbye, I'm going, and then just remove themselves by themselves. They do not need any other enzymes. Group 3 are the most interesting ones, which are called spliceosomal introns. So these need a complex known as a spliceosome, which is a huge RNA protein complex that removes the introns. And finally, group 4 are introns that are required for ATP for splicing. So group 1 and 2, how that occur? So during group 1 and 2, essentially the RNA molecule, there is a particular base that attacks another base. The particular base attacks another base to essentially remove itself, and then there is joining of whatever is remaining. So this can either form from both ends, or it could be one end attacking another, not the other end, but few bases from the other end. So in one case, you form a lariat-like structure. So like this inverted P-like structure, which is known as a lariat. In the other, you just remove a circular strand of DNA, of RNA, and then both the other exons are joined together. So splicing mechanisms of both group 1 and group 2 involve similar steps of two trans-esterification reactions. So first is this joining to this, second is this joining to the other exon, in which a ribose 2' or 3' hydroxyl group makes a nucleophilic attack on the phosphorus and a new phosphodiester bond is formed at the expense of the old one. So essentially, both group 1 and group 2 have the trans-esterification reaction common between them. So during post-transcriptional modification, splicing mechanism of both group 1 and group 2 involve similar steps of two trans-esterification reactions. Okay, any questions? Does anyone have any questions? Okay, moving on. Question 4. Sex determination in Drosophila is a result of blank during post-transcriptional modification. The options are gene amplification, regulated alternate splicing, codon usage change, or nonsense mutation. Which of these are important for sex determination in Drosophila? You can put whatever you think is the correct option in the chat. Jagannath says it is B, regulated alternative splicing. Salum also says it is B, regulated alternative splicing. Anyone else? Chanda and Rupsha also say it is B, regulated alternative splicing. Koel also is saying it is regulated alternative splicing. Bhavna also agrees. Okay, so we have talked about group 1 and 2 introns, which are self-splicing. They do not need anyone to help them. But there is the third category of introns, which are known as which are spliced using a spliceosomal complex. It's a huge protein RNA complex which splices introns. So this is what your gene looks like. You have an exon, and then an intron, then an exon, then an intron, exon, intron, exon. This goes on alternatively, and then you form an immature mRNA in which you have exons as well as these introns present because you have transcribed the entire stretch of the DNA. Next, what happens is these introns are moved out, but during splicing, these either can be moved out completely and you join 1, 2, 3, and 4 to form a big mRNA, or there could be steps known as alternative splicing, in which you join only particular exons and not the others. So you can form a combination such as 1, 2, and 3, shown here, which will form a transcript variant protein A, or 1, 3, and 4, which will form a transcript variant protein B. So you can understand that if this any one of the exons are skipped during alternative splicing, the final protein will obviously have some extra information and some less information. So its function can completely change. So this is known as alternative splicing, in which during splicing, some exons are skipped and others are joined together to form different variants of the same mRNA. So alternative splicing is a method substantially used for many mammalian genes and can result in multiple proteins that are structurally and functionally variable from the same primary transcript. Sometimes alternative splicing is unregulated phenomenon, while others it is strictly regulated. One of the best examples of regulated alternative splicing is sex determination in Drosophila. In Drosophila, there are three genes which are involved in sex determination: sex lethal gene SXL, transformer genes TRA, and double sex gene DSX. Due to alternative splicing, functional genes produced in females and non-functional genes are produced in males. So alternative splicing occurs. So you can imagine if all three of these have a particular exon missing that is required for its function, then whatever you will form is a non-functional copy or a non-functional protein, which occurs in Drosophila males. But in Drosophila females, you get completely accurate splicing so that you have functionally all these three proteins working their normal functions and you produce a female fly. So alternative splicing can occur by two different methods. One, when the poly A or the cleavage sites are present in the primary transcript, and essentially you join different parts together, and this helps in a lot of variability in the final protein. And this is very important in case of antibodies that are being formed from the body. So, just for your information, antibodies that our body produces to fight off infections are proteins that are formed of two different parts, a heavy chain and a light chain. The heavy chain actually comes from very similar looking genes, but due to this alternate splicing, they can produce a variety of antigen detecting ends that can detect various different bacteria. Another example of alternate splicing, which is I think the most interesting example, is a gene known as DISCAM, DSCAM, DISCAM. This particular gene has, I think I remember, close to 50,000 alternative splicing variants. It is a huge gene, and then it has 50,000 variants that can be found from the same gene because it has a lot of different exons can be joined in or not joined in. And this just shows that how important and interesting alternate splicing can be. You can form so many different proteins from the same starting gene. So sex determination in Drosophila is the result of post-transcriptional modification, which is regulated alternative splicing. Any questions? Okay. Next question. In prokaryotic operon system, the expression of prokaryotic operon leads to monocystronic mRNAs, polycystronic ribosomes, polycystronic mRNAs, or single gene transcription. Saloom says it is B, polycystronic ribosomes. Anyone else? Bhavna, Chanda, and Jagannath all say it is C, polycystronic mRNAs. Okay, so let us look at it. Uh, so we have looked at how transcription works as an introduction, and now we'll look at how transcription is regulated in different prokaryotic systems. So we know that a typical bacteria has a very small genome compared to eukaryotes. So uh, E. coli has close to 20,000 base pairs of a genome, which means it doesn't have a lot of genes. But there are particular genes which are known as housekeeping genes. These genes always need to be active for the bacteria to function. So, say genes that are involved in membrane synthesis, genes that are involved in glycolysis, genes that are involved in transcription, say RNA polymerization, all the genes that make up RNA polymerase. All these are known as housekeeping genes, and they need to be always active. So these are known as constitutively active genes or housekeeping genes. But there are going to be genes that are required for specific conditions. Say, for example, if the temperature drops around the bacteria, so it needs some genes which will make sure that the membrane, which is a lipid bilayer, does not freeze, or it sees that there is some toxic antibiotic going around, so it requires a antibiotic resistance gene to start functioning. So some genes, in these cases, their expression is controlled. So they either can be activated when they are necessary, or they are always on and they are inactivated when a particular condition arises. So this is called transcriptional regulation. So we'll look at it. So in prokaryotes, the most common step at which regulation of gene expression occurs is called transcription initiation. Uh, because making a new mRNA is also going to be energetically costly to the cell. So why make a mRNA if you are not going to use it? If a gene only needs to be activated when it is necessary, you do not transcribe it at all. So most of gene regulation occurs at transcription initiation. You do not let the gene be transcribed, because what will you do with the mRNA? You have wasted a lot of ATP to make the mRNA. So instead of, you do not start transcription at all when you are not needing the gene. So it is energetically the most efficient step to regulate gene expression. Transcriptional regulation occurs in steps after initiation, specifically during elongation and termination. So this is when transcriptional. So when we are talking about transcriptional regulation, this is where it is occurs. So initiation, elongation, or termination, and these are done by gene regulatory proteins, which are of two types, either an activator or a repressor. So an activator will obviously start the expression of the gene. So normally it is off and you will turn it on. And a repressor is deactivates the expression of the gene. So normally if the gene is active, then it just stops the transcription level. In the absence of both activator and repressor, transcription is carried out by the RNA polymerase, is called the basal level of transcription. So there's a very low level of transcription that is always going on. The binding of the repressor decreases transcription, so less than basal level, while binding of the activator increases transcription above the basal level. If both of them are present, then normally repressor works better than activator and you have low transcription. So I will give you a bit of information that you would need probably later. Like, this is a very important bit of information. Transcription is essentially regulated as a kinetically determined chemical reaction. What do I mean by that? So, especially talking about both talking about prokaryotes and eukaryotes. So there you have a promoter on which our RNA polymerase can sit and bind and start doing RNA transcription. But the cell is huge, the DNA is huge. How will you control when RNA polymerase goes and binds to that particular promoter? Because there will be thousands of promoters on the entire genome. So essentially, if you can increase the chance of the RNA polymerase landing on the promoter, it will increase transcription or expression of the gene. If you can reduce the probability of RNA polymerase coming close to the promoter, then you will decrease the expression or transcriptional efficiency of the gene. So these are done by transcription factors. Transcription factors are proteins that regulate how well the RNA polymerase can sit on the promoter and do transcription. So as it is written, there are either transcriptional activators, which are transcription factors which sit on particular DNA segments known as enhancers. and they seek out RNA polymerase subunits and cause them to come together onto the promoter and help stabilize it so that RNA polymerase can make the entire transcription. The other kind is called a transcriptional repressor. These are transcription factors that block RNA polymerase from sitting on the promoter. So normally, there is a basal level of transcription, which is probabilistically very, very low because there are thousands of promoters that the RNA polymerase can bind. Your particular gene XYZ, it is not specifically binding to that. There is no force to make it bind or not make it bind. So there is a very low level of transcription always occurring. When you need to improve the level of transcription, you use a transcriptional activator, which is a transcription factor which will sit on the enhancer for that gene and essentially catch hold of RNA polymerase and make it sit on the promoter so that you increase the efficiency of transcription. If you want to downregulate the gene, you use a transcription repressor, which is a transcription factor that can again bind to enhancers, which are gene segments close to the gene of interest, and it will prevent the RNA polymerase to either sit on the promoter sequence or it will cause so that the RNA polymerase is not stabilized, so it can not do transcription at all. So activator and repressor and enhancers are the segments on which activators and repressor will come and bind. Okay, so the basic concept of how gene regulation occurs at transcriptional level in bacteria was provided by the classical model known as the operon model. So this is what we are going to discuss now. So what is a operon? Operon is a genetic regulatory system mostly seen in prokaryotes, in which there are a number of genes that are transcribed together for the same promoter. And this entire thing is known as the operon. There is also the presence of a DNA segment known as the operator, on which an activator or a repressor can sit and bind. So essentially, a polycistronic gene which can be regulated using an operator sequence is known as an operon. As we have said that prokaryotic operons are polycistronic. So the difference is the expression of prokaryotic operons leads to polycistronic mRNAs, meaning you will transcribe this entire stretch, but each of these will form a different mRNA. So you will have mRNA A, mRNA B, mRNA C. Whereas in eukaryotes, if you transcribe something, it is going to form one mRNA only, which is going to be mRNA A. It might be spliced to form different transcript variants, but they are never going to be different mRNAs completely, which will form different proteins. It only occurs in case of prokaryotes. So prokaryotic operons leads to polycistronic mRNAs, while eukaryotic operons lead to monocistronic mRNAs. So in prokaryotic operon system, the expression of prokaryotic operon leads to polycistronic mRNAs. Any questions? Okay. Okay. The repressor or activator which controls the operon system is not part of operon because blank. It codes for enzymes, has its own promoter, it is not coding region, it operates independently of the operon. Mm, Saloom says it is D, it operates independently of the operon. Chanda says it is B, it has its own promoter. Anyone else? Jagannath says, and Ruksha also say it is B, it has its own promoter. Okay, let us look at it. So we talked a bit about operon. So this is a general structure of a operon. So this is the part that we just saw. So we have a promoter on which our RNA polymerase will bind. There is an operator DNA sequence on which our activator repressor molecules bind, and it is generally in between the promoter and the rest of the transcriptional unit. and then we have the structural genes which will form different proteins. But all of this is also accompanied by a regulatory gene. This regulatory gene is a is present is could be present far away from the operon, but essentially it forms a regulatory protein, which is either the activator or the repressor, which will come and sit on the operator. So the regulatory gene is transcribed by a different set of promoter, and normally it forms an activator or repressor, which will come and bind on the operator of the operon we are interested in. So this is completely on the other side somewhere, and this is where it comes and acts on. So this kind of element, which is not present in the vicinity normally, but comes from somewhere else and helps in expression of a gene, either regulation by down regulation or up regulation, is called a trans-acting element. A trans-acting element is a factor which is present somewhere else. It comes from somewhere else and then interferes with our expression of genes. So a trans-acting element. Then there are cis-acting elements which are present very close by. So for example, promoter is a cis-acting element. So it is present close by and it actually helps in transcription of the gene. Operator is a cis-acting element, which is a same DNA, same very close DNA segment, which helps in regulation of the gene. Now, as for the regulatory gene, it codes for a protein called a regulatory protein, which could either be a repressor or activator, but it is not part of the same operon system because it has its own promoter. So there will be a promoter right here for the regulatory gene, and this entire cassette can be somewhere else in the entire genome. So it has its own promoter. So the repressor activator, which controls the operating operon system, is not part of the operon because it has its own promoter. So this is the reason it is not part of the operon. It is not that it acts independently of the operon. So we will see examples in which there are molecules which will help in regulation of our gene, but they are completely independent from the gene itself. So that is not our repressor or activator. Repressor or activator is just not part of the same DNA locality as our operon system. It has its own promoter and it will form a protein that will take part directly in the operon mediated regulation. So operator is a blank and acts as a site of repressor attachment. The options are promote protein complex, segment of DNA, binding site for RNA pol, and ribosomal subunit. Chandra says it is B, segment of DNA. Jagannath also says it is B, segment of DNA. Anyone else? Koel and Ruksha and Salum and Bhavna all say it is B, segment of DNA. So let's look at it. So we have just seen operator is a segment of DNA which is in between the promoter and the structural genes on which our repressor or our activator can come and bind. So essentially it is a segment of DNA. That is just it. So promoter is a nucleotide sequence that acts as a site for RNA polymerase attachment and transcribe a gene. RNA polymerase recognize the promoter and then initiates transcription. Operator is a segment of DNA and acts as a site of repressor attachment and generally situated between the promoter and the structural genes of the operon. And the structural genes are present at the end of the operon, and these genes are transcribed to a single mRNA and translated into various different proteins. So a polycystonic mRNA, one mRNA to different proteins. Okay, operator is a segment of DNA and acts as a site of repressor attachment. So one of the operons that we are going to discuss is called the lac operon. The lac operon contains three structural genes which are blank. The options are lac A, lac B, lac Z, lac C, lac Z, lac Y, lac A, lac X, Y, and Z, lac R, T, and Y. You can choose what you think is the correct option in the chat. Okay, we have answers coming in. Jagannath and Salum say it is B. The lack of brain contains the structural genes like Z, like Y, and like A. So yes, we are going to talk about this. So one of the basic Bhavna also says it is B. One of the basic and most studied operons is known as the lactose operon, which is short form is lacoperon. So lacoperon in E. coli contains genes which are required for the lactose metabolism. So lactose is obviously another sugar like glucose, but lactose produces way less energy and requires way more proteins to be utilized to produce energy. So normally E. coli always will prefer glucose if it is present. But if glucose is present, if glucose is absent and lactose is present, then E. coli would start transcribing this particular operon to for use the proteins for utilizing lactose as its energy source. So what does the lac operon have? Lac operon has a promoter, it has an operator, it has three structural genes, LAC-Z, LAC-Y, and LAC-A. These are beta galactosidase, beta galactoside permease, beta galactoside transacetylase, essentially everything you need to use lactose as the energy source. Then there is a cap site, we will see what that is. And then we have a promoter for LAC-I or LAC inhibitor, which is the repressor for this entire operon. We will see how it works. So the lacoperon contains three structural genes like Z, like Y, and like A. These genes are transcribed as a single mRNA under the control of one promoter and will be then during translation form three different proteins. The lacoperon is typically shut off or repressed in normal conditions, but can be activated in the presence of lactose, which is converted to allolactose. Thus, the lacoperon is an inducible operon. So when you have glucose in plenty, you do not need the lactose metabolism gene. So that is just a waste of energy. So normally it is shut down heavily. But when you have a lack of glucose, it is activated and thus it is known as an inducible operon. It can be induced or it can be switched off, switched on. Normally it is off. So what are the different parts of the lactose operon? The structural genes like Z, which is beta galactosidase, which converts lactose into glucose and galactose, which can go directly into glycolysis. And this enzyme also converts lactose to allolactose. Likewise, beta galactosidase permease, which transfers lactose into the cell, and LAC-A helps in detoxification. And the regulatory gene sequence has a promoter, has an operator, and also has a cap binding site. So, what is cap binding site? The cap binding site is a positive regulatory site bound by the catabolite activator protein or cap. When cap is bound, it promotes the RNA polymerase to do transcription. And LAC-I is the repressor that just binds to the operator and prevents transcription. So the lacoperon contains three structural genes, LAC-Z, LAC-Y, and LAC-A. When lactose is not available, the lac repressor blank with the operator of the lacoperon. Weakly binds, strongly binds, dissociates, or loosely dissociates when lactose is not available. Salum, Chanda, and Koel have said it is option B, which is strongly binds. Anyone else? Jagannath and Rupsa also said this is B, strongly binds. So we will now discuss how exactly the lacoperon works. So So we have already seen this. The lac operon has a cap binding site, a promoter, a operator, a lac Z, Y, and A, which are the structural genes. So the first condition is glucose is absent, but lactose is that should not. Yeah, we will go it in the opposite direction. Yeah. So first thing is glucose is present and lactose. No, we should go. I will. We will go in the correct direction. Glucose is absent and lactose is present. So when glucose is absent and lactose is present, you want maximum transcription from this operon. So what happens is we have our lac. Mm, no, I don't like this. We will start here. When glucose is present and lactose is absent. So we have seen that we have a repressor, which is this. This is our repressor. And when glucose is present and lactose is absent, our repressor gets produced. It sits on the operator and prevents RNA polymerase to do any transcription. So essentially, you have no transcription when glucose is present and lactose is absent. Next, when glucose is present and lactose is also present, in this condition, lactose is converted to one of its allosteric forms or isomeric forms, which is called allolactose. Allolactose goes and sits on the repressor and prevents it from binding to the operator. So when both glucose and lactose is present, there is low level of transcription of lactose metabolism gene because the cell is like we can use a bit of transcription, but it is not very good transcription. So because glucose is present, you know, need not a lot of lactose metabolism. So there is low or basal level of transcription of lactose metabolism genes. Next, when glucose is absent and lactose is present, so we have the same thing where allolactose goes and binds to the repressor. So the operator is empty. RNA pol can bind and do its transcription. But in the absence of glucose, something else happens as well. The cells, which is essentially bacteria such as E. coli here, they produce cyclic AMP. Cyclic AMP is like a hunger signal. The E. coli is saying that we do not have enough ATP, so it forms cyclic AMP. Cyclic AMP binds to this cap protein, which we have seen the catabolite associated protein. So it cyclic AMP binds to cap protein and activates it. When it is activated, this complex sits on the cap binding sites and activates RNA polymerase. So essentially, it enhances the expression of the entire operon. So it causes RNA polymerase to come and sit on the promoter, essentially causing high levels of transcription. So it acts as a activator or an enhancer for the lac operon transcription in this case. So glucose absent and lactose present, we have high levels of transcription. And finally, when both are absent, then even though the cell produces cyclic AMP, the cap and cyclic AMP both are activated and sit on the cap binding site, RNA polymerase is recruited, the operator is still repressed by the repressor protein because it sits on the operator. So RNA polymerase cannot walk on the operon. So essentially, there is no transcription again because lactose is absent and you do not need lactose metabolism genes already when you do not have much energy. So these are the four conditions. First, glucose present, lactose absent. In that case, repressor prevents any transcription because glucose is present. When both are present, then repressor is removed. You have low levels of transcription. When glucose is absent and lactose is present, you have a CAMP cap binding signal, which causes high levels of transcription because it recruits the RNA polymerase actively and causes high levels of transcription of the lactose metabolism because the cell needs energy and glucose is not there. So lactose metabolism can be ramped up as far as possible. And finally, when both are absent, the cell is in a low energy state. It does not have any food, then even though it is producing the CAMP hunger signal binding to cap and the cap is binding to cap binding site, RNA polymerase is actively being recruited, but due to the absence of lactose, repressor binds to the operator and does not allow any transcription. So this is how the entire lac operon functions. So glucose, if it is present, lactose absent, no transcription. Both are present, low transcription. Both are absent, no transcription. And glucose is absent and lactose is present, high levels of transcription. So when lactose is not available, the lac repressor strongly binds to the lac operon. Any questions? Does anyone have any questions? Okay, moving on. In lac operon, blank acts as a inducer. Lactose, allolactose, glucose, or galactose. Saloon says it is B. Allolactose that acts as an inducer. Anyone else? Ruksha, Jagannath, and Chanda all say that in lacoperon, allolactose acts as the inducer. So let us look at it. So this goes through the same thing that we have just discussed. Uh, so when lactose is not available, then repressor binds to the operator and RNA polymerase cannot transcribe it. So there is no transcription from the lacoperon. But when lactose is present, allolactose is an isoform of lactose that is formed by beta galactosidase, and it binds to the operator and causes it binds to the repressor and causes it to move off the operator. And now RNA pol can bind and produce essentially transcribe the lacoperon. So there are various other ways of doing it. So not only allolactose, we have found other chemical analogs for allolactose which can relieve the repressor from the operator. These are IPTG, phenylgal, TMG. So these are chemicals that if we put on the cells, they will think there is a lot of lactose present and they will start transcribing the lacoperon. This particular trick has been used to make very clever systems uh, which are used in plasmids a lot in molecular biology, and we'll look at how that works. So in lacoperon, allolactose acts as the inducer. Next, high amounts of lacoperon transcription are only possible without lactose, fructose, galactose, and glucose. High levels of transcription will only be produced in the absence of what? Saloom and Chanda say it is D. Ruksha also say it is D. Glucose. Jagannath also says it is D. Glucose. So you're all correct. We have seen that when lactose is present and there is no repressor binding, still the level of transcription will be low if there is too much energy in the cell. If there is no glucose present, then the E. coli produces a CAMP as the hunger signal and it binds to cap protein. Cap protein is then activated, it binds to the cap binding site in the absence of glucose and it actively recruits RNA polymerase, essentially increasing the transcription rate of the entire lac operon. So high levels of transcription occurs in the lacoperon in the absence of glucose. The high amounts of lacoperon transcription are only possible without glucose. Next, we are going to talk about the other operon, which is the tryptophan operon. And tryptophan operon is blank when tryptophan concentration is high in the environment. So, what does the tryptophan operon do when there is very high amounts of tryptophan around it? So, tryptophan operon essentially produces tryptophan. So, what are the following it does? It becomes inactive, it becomes active, it becomes repressed, or it becomes induced. Okay, Chanda, Saloom, and Jagannath all say it is C. It becomes repressed in the presence of tryptophan. Let us look at it. So we are going to look at tryptophan or the trypoperon. The trypoperon found in E. coli bacteria and it encodes genes for production of tryptophan. Normally, it remains on in case of low tryptophan and off in the presence of tryptophan. So tryptophan does not need to be synthesized E. coli when it is present in the environment. Hence, transcription of genes of trypoperon are off. On the other side, when the availability of tryptophan is low, the operon becomes on and the genes are transcribed. Trip repressor, which is the repressor for the trip operon, does not always attach to DNA. Instead, it binds and inhibits transcription only in the presence of tryptophan. So normally, the tryptophan operon essentially is on, and when there is a lot of tryptophan, it turns it off. Tryptophan binds to the repressor molecules and alters the structure, and we'll see how that happens, and it is known as a co-repressor. And lac operon, we have seen, works by inhibiting the initiation of transcription, like most bacterial operons. But in case of tryptophan operon, it does something a bit different. One unique feature of tryptophan is known as attenuation. So, like regulation by trip repressor, attenuation is a mechanism for reducing expression of tryptophan when the levels of tryptophan are high. So it represses, but it does not block the initiation of transcription as we have seen with lacoperon, but it prevents completion of transcription. So elongation is prevented by tripoperon. So it does not block initiation but blocks elongation. And we will see how that works. So this is the structure of the tripoperon. So tripoperon has a promoter, has a operator sequence. Then it has the structural gene strip A, B, C, D, E in the opposite direction and also has a trip L, a trip leader sequence, which helps in attenuation. And finally, it has a independent promoter that drives the trip repressor. We will see, so it contains the trip repressive regulatory gene trip R. Trip R by in the presence of tryptophan, trip R binds to the operator blocking transcription of tripoperon by inhibiting RNA polymerase binding, very similar to LAC inhibitor that we have just seen. So, but we are more interested in the attenuation factor by trip L in this case. But trip R acts very similarly to how LAC I works in lacoperon. It is a repressor that is formed by its own independent promoter. In the presence of a lot of tryptophan, TRPR is activated. So normally it is in inactive state, a lot of tryptophan is present, becomes activated and it goes and sits on the operator preventing initiation of transcription of the TRP operon. Because if there is a lot of tryptophan, you do not need transcribing of the TRP biosynthesis pathway. So, so how is tryptophan operon regulation works? In absence of tryptophan or low tryptophan, when there is little tryptophan or absence of tryptophan, even if the trip repressor is expressed, it is expressed in an inactive form, and thus it cannot do anything. And RNA polymerase binds and transcribes the entire trip operon, and we have trip biosynthesis enzymes, and you make more tryptophan. But In the presence of high levels of tryptophan, when the TRPR enzyme is formed, so inactive repressor is formed, tryptophan binds to this repressor and forms an active repressor. And this active repressor goes and sits on the operator and prevents any more biosynthesis or like synthesis of the tryptophan operon, and thus you do not make any more tryptophan producing enzymes because there is already a lot of tryptophan in the system. So transcription is repressed in the presence of lot of tryptophan. So tryptophan operon is repressed when tryptophan concentration is high in the environment. So everyone was correct in their option. Next, in tryptophan operon, attenuation blank enhances transcription, terminates translation, reduces mRNA stability, or prevents completion of transcription. So what does tryptophan operon do? Like what is attenuation out of these following four options? You can choose whatever you think is the correct option. Okay, Chanda, Jagannath, and Salum also it is D, prevents completion of transcription. So let us look at how this works. So this is a bit complicated. So you will have to pay a bit of attention here. So uh, whenever we are talking about a repressor binding to the operator sequence and repressing transcription, we have to understand that that does not mean that transcription is absolutely stopped. It means the transcription is very like reduced by a lot, but there is always something known as leaky transcription. Genes are always transcribed even though the operator tries its best to prevent it. The repressor tries its best to prevent transcription. Transcription can still occur. So the tryptophan operator, in addition to having the a tryptophan repressor binding to the operator sequence, has a second mode of repressing the operon itself when there is high amounts of tryptophan. So a second mode. This is known as attenuation. So first we have to understand how the entire operon is laid out in front of us. We have the promoter, we have the operator, then we have a trip L sequence. This is the leader, the tryptophan leader sequence. And then after that, we have EDCBA, all the structural genes that are present in the polycistronic mRNA, and these are the actual ones that are required for tryptophan biosynthesis. Now, when there is a high level of tryptophan, when the operon needs to be down repressed, then in that case, the leader sequence is first transcribed and it prevents the transcription of anything downstream. But if there is a low tryptophan leader sequence does not do anything like that. It lets the entire thing be transcribed, and we are going to see how that occurs. So this all lies in how the tryptophan leader sequence is made. The tryptophan leader sequence has four regions: region 1, region 2, region 3, and region 4. So this is we are only talking about this red region, okay? So it has region 1, 2, 3, and 4. So the function of the leader sequence is to fine tune the expression of the tryptophan operon based on availability of tryptophan in the cell. The In the region 1, the unique factor about it is that it has two tryptophan codons, which means when it is being translated, it would need tryptophan. And the translational stop codon lies between regions 1 and 2. This will come in handy later. Further, uh, region 2, 3, 4, which are the next three regions, the region 3 is complementary to both region 2 and region 4. If region 3 and 4 base pair with each other, they form a loop-like structure called the attenuator and functions as a transcriptional termination. If the pairing occurs between region 3 and 2, then no such attenuation occurs and transcription is continued. We see it using this particular example. So tryptophan leader sequence, which is 1, 2, 3, and 4. Now consider the case when there is low amounts of tryptophan. So our RNA polymerase has sit on this end and has transcribed the region 1 and is going forward towards region 2. Ribosome, which we have seen that the translation occurs at the same time. So ribosome will also sit on the newly made mRNA and keep on translating as RNA polymerase keeps going downstream. Now, ribosome at one point will reach the two tryptophan codons that are present in region 1. In case of high levels of uh, tryptophan, in case of high levels of tryptophan, uh, the ribosome will not stop at this stage and will continue till it reaches region 2. And there is a stop codon between region 1 and region 2, and the ribosome will stall here. Essentially, it will stop here. And it covers part of region 1 and part of region 2. We have seen that the region 3 is complementary to both region 2 and region 4. If the ribosome stops at region 2, the region 3 is free to do complement, like form a hairpin loop between 3 and 4. This complementary loop we have seen during non-row dependent or row independent transcription stop or termination. We have seen such a hairpin loop causes the RNA polymerase to fall off. So essentially, this is what happens when there is a lot of tryptophan. The ribosome just zooms past this stage and stops only at the stop codon between 1 and 2. This causes 3 and 4 to form a hairpin loop, and RNA polymerase always treats this as a transcriptional termination site and falls off. So essentially, anything after region 4 is not transcribed. So essentially, everything after the trip leader is not transcribed, so everything here stops. So elongation of the mRNA stops. So this is how a leader causes repression of the operon when tryptophan levels are high. But on the other hand, if tryptophan levels are low, the same thing occurs. Our RNA polymerase forms the mRNA for 1, 2, 3, and 4 and is continuing downstream as the ribosome sits on the newly made mRNA and starts translating it to a protein. But if there is low levels of tryptophan, then when the ribosome reaches this mRNA sequence of when it reaches this mRNA sequence of two tryptophans, then it will completely stop here, only in the region 1. And in this case, region 2 is now free to form a loop with region 3. But when 2 and 3 form a stem loop, by this time the RNA polymerase is already at region 4. And thus it is not stalled, and it essentially has never seen the transcription termination site because it has already moved to 4 by this time, and it completes the transcription of the entire operon. So however, if the tryptophan is in short supply, the ribosome will pause at two tryptophan codons contained within the sequence 1. This leaves sequence 2 free to base pair with sequence 3, forming a 2-3 structure called an anti-terminator. 3,4 cannot form the transcription stop site, and thus transcription by RNA polymerase continues till the end of the trip operon. So in trip operon, attenuation prevents the completion of transcription. Any questions? Okay. The three structural genes RRB, RRA, and RRD are found in which are the following: lac-operon, gal-operon, arabad-operon, or trip-operon. You can put what you think is the correct option in the chat. Jagannathan and Salum say it is C, arabad operator. Anyone else? Okay, Jagannathan and Salum say it is C. No one else seems to know the answer, so we'll look at it. So this is the last operon we'll talk about. This is called the arabinose of operon. So arabinose is a 5-carbon sugar, which is used to make a lot of other sugars and downstream molecules, and it is present as a the ara operon, and we will see how ara operon is works. So this is what ara operon looks like. It's a bit more complicated than the other two. So we have ARA-C, ARA-O2. ARA-C is the regulatory gene, which in this case is transcribed by the same promoter, known as para-C, uh, which is transcribed by the same promoter, which is very close, called para-C. And then we have another ARA-O1 gene, which is also transcribed by para-C. Then we have the cap binding site, ARA-1, ARA-2 sites. Then we have this pair ABD, which is the promoter for the structural genes ARA-A, and ARA-T. So Aravinose operon is composed of three structural genes and regulatory regions, with the operator regions ARA-O1 and ARA-O2. So there are two different operators, O1 and O2, and the initiator regions I1 and I2. These two, we'll see how that works. And the structural genes are BAD. There is also a cap binding site where the cap cyclic AMP complex binds and facilitates catabolite repression and results in positive regulation of ARA-BAD when the cells lack glucose. The regulatory gene ARA-C is located upstream of L-arabinose operon and encodes arabinose responsive regulatory protein ARA-C. This is our regulatory or repressor protein ARA-C. Both ARA-C and ARA-BAD have a specific promoter where RNA polymerase binds and initiates transcription. Arab-Bad and Arab-C are transcribed in opposite directions from the Arab-Bad promoter, PBAT, and Arab-C promoter respectively. So one transcribed this way and one this para ABD transcribes this way. So three structural genes in Arab-B, A, and D are found in the Arab-Bad operator. Final question for today. The L-arabinose system is positively or negatively regulated by the binding of the blank cap protein, lac repressor, GCN4 protein, or ARA-C protein. Jagannath and Chanda Saloom all say it is D. It is RC protein. You're all correct. So let us look at it. So we have the CAP-CAMP activator as in the lac operon, but on top of that, we have ARA-C, which is a regulator. So it can act both as a positive or a negative regulator for the ARA operon. So when there is arabinose, it undergoes a dimerization, and when there is too much arabinose, it causes downregulation, such as this. I will just show you here. So ARA-C is this yellow figure right here. When there is a so when there is no arabinose, then we do not need the arabinose metabolism gene. So ARA-BAD, they metabolize arabinose to other useful sugars. So when there is no arabinose, you do not need any arabinose metabolism genes. So in that case, when ARA-C is formed, it actually binds to ARA-O2 regions present both upstream and downstream, causing a looped DNA structure such as this. This looped DNA structure essentially causes steric inhibition, which prevents the RNA polymerase to bind to the promoter of ARA-BAD. Essentially, then in the absence of arabinose, no transcription takes place. But on the other hand, when there is a hunger signal CAMP and CAP is activated to bind to the CAP binding protein, this entire region opens up. When it opens up and then there is a lot of arabinose binds to the ARA-C molecules and causes it to act as an activator by binding to the ARA I1 and ARA I2 regions. This entire process causes activation of RNA polymerase to sit on the promoter and cause transcription of the gene. So no arabinose, then the DNA loops around, does not let RNA polymerase sit. In the presence of cyclic AMP and a lot of arabinose, the DNA opens up and promotes sitting of RNA polymerase at the promoter region and transcription of the metabolism related genes. So L-arabinose is positively or negatively regulated both by ARA-C protein, depending on presence or absence of arabinose. Okay, so that is all I had for you today. Any questions anyone has? Okay, if everything is fine, then thank you for joining everyone today. I will see you next week. Bye.